So, if you look at the breast tissue of a completely healthy, cancer-free 50-year-old under a microscope, you might think everything is perfectly fine.
Right. It looks completely intact.
Exactly. There are no tumors, no malignant masses, but at the molecular level, her cells might be secretly rolling out like a biological red carpet for a tumor that doesn't even exist yet.
It's just terrifying, honestly.
It really is. I mean, we always hear that aging is the absolute biggest risk factor for breast cancer. But the assumption is usually just that our cells get old, they get tired, and, they make a mistake.
Yeah, which is a very passive way of looking at it. The reality of aging tissue is aggressively active. It's not just a slow decline. It is a highly orchestrated, localized rewriting of the tissue's fundamental rules.
And that is exactly what we are exploring in today's deep dive. We have a groundbreaking study from Nature Aging that completely reframes how we think about risk.
Yeah, the researchers built what they call a single-cell aging atlas.
Right, so they took completely healthy mammary glands from young 3-month-old mice and compared them to older 18-month-old mice. Their goal was to find out exactly how aging pre-wires the tissue for cancer.
And the way they did this involves two incredibly powerful sequencing technologies that we really need to define right up front: scRNA-seq and snATAC-seq.
Okay, let's break those down because they are a mouthful.
They are, but they're really the lens that makes this entire study possible. First, scRNA-seq, or single-cell RNA sequencing. This allows us to isolate every individual cell in the tissue and read the exact RNA transcripts it is actively producing at that exact millisecond.
So it tells us what the cell is currently doing right now.
Exactly.
Right, so to visualize that, for you listening, if a cell is a musician, the scRNA-seq is us looking directly at the sheet music they're currently playing.
That's a great analogy. But it's the second technology, the snATAC-seq, that really gets to the root of the aging process.
Because it looks at the structure, right?
Yes. So snATAC-seq stands for Single Nucleus Assay for Transposase Accessible Chromatin. Wow, okay.
I know, it's a lot. But basically, instead of looking at the RNA being produced, we are looking at the DNA itself, specifically its physical structure. Our DNA is incredibly long. So to fit inside a cell, it's tightly spooled around proteins called histones, kind of like thread on a spool.
Okay, I'm picturing that.
That whole structure is called chromatin.
And if the DNA is tightly wound up, the cell's machinery physically cannot reach it to read those genes, right?
Exactly. The snATAC-seq maps out epigenetic accessibility. It shows us which sections of the DNA thread have been unspooled and left open to be read, and which sections have been tightly wound and essentially locked away.
Okay, so keeping with our musician analogy, snATAC-seq is looking at the piano itself. It tells us which keys have been permanently superglued down so they can never be pressed, and which keys have had their covers stripped off so anyone can just smash them.
Yes, perfect. It defines what chords the cell is even capable of playing.
So cool. And when the researchers applied these two tools to the epithelial cells, the workers that actually form the inner ducts and lobules of the mammary gland, they found a workforce undergoing a massive identity crisis.
They really did. Let's start with the physical shift. In a young mammary gland, you have this beautiful balance.
Right.
There are luminal cells that line the inside of the ducts, and there are myoepithelial cells that surround the outside.
And those outer cells act almost like a structural corset, right, to keep everything contained.
Exactly. But as the tissue ages, the luminal cells decrease in proportion, and the myoepithelial cells dramatically increase. But the mechanism here isn't just about cell death and cell division. It's about lineage fidelity loss. These cells are literally forgetting their biological roles.
Yeah, they're forgetting their jobs. How does a cell just, forget what it's supposed to do?
It comes down to those piano keys you mentioned, the epigenetics.
Oh, okay. The myoepithelial cells, for example, begin to tightly wind up and shut down the chromatin around their defining identity markers, specifically genes like KCARTA17 and KAT5.
So the keys get glued shut.
Yes. The cells are still physically sitting in the tissue, but they are shutting off the genetic instructions that make them behave like proper myoepithelial cells.
That is wild. And the luminal cells inside the ducts, are they also losing their grip on reality?
Very much so. And this is where the link to cancer risk really begins to crystallize. The researchers tracked a specific gene in the luminal cells called FNDC4.
FNDC4, what does it do?
Well, in a young, healthy cell, FNDC4 produces a crucial anti-inflammatory protein that keeps the tissue environment calm. But with age, the promoter region for FNDC4, the on switch, basically becomes epigenetically inaccessible.
You get glued shut, too.
Right.
So the cell physically loses the ability to calm down the tissue. It just drops its anti-inflammatory shield.
Exactly.
But if the shield goes down, what's coming up to replace it? Something has to fill that void.
Well, the chromatin opens up around a gene called HP, which produces haptoglobin.
Okay. And why is that bad?
Because this gene is heavily implicated in metabolic reprogramming. It alters how the cell processes energy, and it is frequently associated with the development of breast cancer.
Oh, wow.
Yeah, so the luminal cell is not only forgetting its normal job, it's actively shifting its metabolism toward a cancer-friendly state.
Wait, I want to pause here because looking at the data, there is a serious paradox here.
Oh, the tumor suppressors.
Yeah. While they're turning on metabolic genes like HP, the age luminal cells also start unspooling the DNA to upregulate AB41L3.
Right.
But AB41Ep3 is a known tumor suppressor gene. Why would a cell that is seemingly drifting toward a cancer state suddenly just hammer the tumor suppressor button?
It's fascinating. It reveals a profound internal struggle within the cell.
Like a tug of war.
Exactly. The cell is not completely oblivious to the chaos. It detects the rising inflammation. It senses the abnormal metabolic shifts caused by the HP upregulation.
So it's trying to save itself.
Yes. In response to this mounting internal stress, transcription factors, which are proteins that turn genes on, like the famous tumor suppressor TP53, become highly active. They are desperately trying to stabilize a destabilizing genome.
Okay, so let me just make sure I'm capturing this whole picture of the epithelial breakdown for you listening, just to give a quick recap of this first section.
Go for it.
First, the physical balance of the tissue flips, with luminal cells dying off and myoepithelial cells taking over. Second, the cells undergo an epigenetic identity crisis where they shut off their specific job markers. And third, the luminal cells become an active war zone, turning off their anti-inflammatory shields while simultaneously slamming on their internal tumor suppressors to fight off the metabolic chaos.
That is exactly the dynamic, perfectly summarized. But here is the critical next step in the mechanism. These struggling epithelial cells do not exist in a vacuum.
Right, they have neighbors.
Exactly. As they lose their identity and experience this immense internal stress, they begin secreting abnormal chemical signals into the surrounding environment.
And that environment is the stroma, right? The physical connective tissue and an infrastructure that holds the whole gland together.
Yep. And the primary builders of this infrastructure are the fibroblasts.
Okay, so let's talk about the fibroblasts.
So fibroblasts are the architects of the extracellular matrix. They lay down the collagens and the structural proteins that give the breast tissue its shape and structural integrity.
So they're the construction workers of the city.
Great way to think about it. And when they are bombarded by the distress signals from the aging epithelial cells, their behavior changes radically. The total number of fibroblasts surges, but one specific subtype, known as FibC2, increases sixfold.
A sixfold expansion, that's massive.
It is.
But they aren't building normal tissue anymore, are they?
No, they are not. They enter a state of senescence. Now, senescence is often casually described as a cell retiring or going to sleep, but that is a dangerous misconception.
Right, they aren't just napping on the job.
Not at all. A senescent fibroblast is highly active. It has simply stopped dividing and instead morphed into a hypersecretory zombie state.
Okay, a hypersecretory zombie state. I love that phrasing. But what exactly is it secreting?
Well, a young, healthy fibroblast primarily secretes a protein called fibronectin, coded by the gene FN1. This provides a smooth, organized scaffolding.
Like a well-ordered suspension bridge.
Yes, but the senescent FibC2 cells entirely start making fibronectin. Instead, they activate genes like col6A3 to start pumping out unusual, disorganized collagen.
So they're essentially replacing that suspension bridge with a chaotic, tangled web of rebar.
Exactly. And a tangled web of structural proteins is exactly what a tumor needs to anchor itself and grow.
Wow.
But the gene expression of these fibroblasts goes even further than just building bad scaffolding.
It does.
Yeah. These aged senescent fibroblasts begin upregulating CDKN1A and CDKN2A, which are classic markers of DNA damage and permanent cell cycle arrest. But the most alarming discovery was the massive upregulation of a gene called FAP.
Wait, FAP? FAP? Right. Because in oncology, FAP is the defining marker for a cancer-associated fibroblast, or a CAF.
That's right.
If this mouse is 100% healthy, and there are absolutely zero tumor cells in its body, how is a cancer-associated marker getting turned on at all? That seems like a massive biological error.
It highlights how chronic tissue stress mimics the exact environment of a growing tumor.
Oh, I see.
A tumor is essentially a wound that does not heal. It secretes constant inflammatory signals that hijack normal fibroblasts, turning them into C-halves that feed and protect the tumor.
Right.
What this study proves is that the sheer stress of the aging process, the identity loss of the epithelial cells, and the resulting inflammation we just talked about is enough to trigger that exact same hijacking.
So the aging process is tricking the fibroblasts into believing there is already a tumor there that needs to be supported.
Precisely. They also upregulate genes like FABP4, which is responsible for transporting fatty acids.
Okay, and tumors love lipids. Right.
They do. Tumors require massive amounts of lipids for energy to fuel their rapid growth. The aged fibroblasts are literally preparing a lipid-rich fuel supply in the surrounding tissue.
So essentially, before the first cancer cell even mutates, the surrounding tissue has already built its home and stocked its fridge.
That's a terrifyingly accurate way to put it.
Okay, so tracking the cause and effect here for you listening, recap number 2. The epithelial cells get confused and send out distress signals. The fibroblasts panic, multiply, and turn into senescent zombies. They stop building smooth tissue, start building tangled, tumor-friendly scaffolding, and turn on genes that hoard fatty energy, creating a perfect soil for cancer.
Exactly. Which brings us to the final layer of defense.
Right, the immune system. Because if the epithelial cells are compromised and the fibroblasts are building a corrupted infrastructure, the immune system should theoretically step in, recognize the senescent cells as abnormal, and clear them out.
That's what you would hope.
Right. So why aren't the immune cells patrolling this area doing their job?
Because the immune microenvironment undergoes an equally profound age-driven transformation.
Of course it does.
In young tissue, you have a vast reserve of naive T cells. These are undifferentiated, highly flexible immune cells just waiting to be presented with a new threat.
Like fresh recruits.
Yes. But in the aged mammary gland, the naive T cells drop drastically.
And they are replaced by a massive expansion of memory T cells, right? Looking at the source, it specifically highlights CD4 plus memory cells, gamma delta T cells, and a very specific group of CD8 T cells known as GNK plus T cells.
Yes. Identifying these cell types is crucial, but what makes this study groundbreaking is where they found them.
Ah, the spatial mapping.
Right. The researchers used a technique called spatial transcriptomics. Instead of just grinding up the tissue to see what genes are there, they slide a completely intact slice of the tissue onto a specialized microchip to map the exact physical location of every single RNA transcript.
It's like moving from just having a list of ingredients to having an actual GPS map of the tissue.
Exactly. And the GPS map revealed that in young mice, these immune cells largely remain sequestered in the lymph nodes, waiting to be called upon.
Staying at the base.
Right. But in the older mice, these GZMK plus memory T cells physically migrate out of the lymph nodes and infiltrate the mammary ducts. They swarm the aging epithelial cells.
So they see the threat, they physically swarm the confused epithelial cells. But if they are swarming the area, why aren't they clearing out the dysfunction?
To figure that out, the researchers ran the single cell data through a computational algorithm called cellphone-DB.
Cellphone-DB, like a database of cell phone calls.
Kind of. It's a tool that looks at the surface proteins produced by one cell and the receptors produced by a neighboring cell, and it infers literal molecular conversations between them.
Oh, that is so cool.
It really is. And it turns out the immune cells aren't attacking the epithelial cells. They are being actively suppressed by them.
Okay, how does that physical suppression actually work? Walk me through the mechanism.
It operates like a corrupted lock-and-key mechanism. The aged, confused epithelial cells dramatically upregulate a gene called FAM3C, which produces a protein that sits on the outside of the cell.
Okay, the FAM3C key.
Right. At the exact same time, the swarming GZMK plus T cells begin expressing incredibly high levels of a receptor called PDCD1.
And PDCD1 is the gene for PD-1.
Yes, it is.
That is one of the most famous exhaustion markers in immunology. When PD-1 is triggered, it essentially forces the immune cell to stand down and go to sleep.
Yes. The cellphone-DB mapping showed that the FAM3C protein on the aged epithelial cell perfectly docks with the PD-1 receptor on the T cell. The aging duct cell is actively reaching out, inserting a key into the immune cell's lock and shutting it off.
Oh my God. So the immune failure isn't passive. It's not just that the T cells are too old and tired to notice the mutated fibroblast. The aged tissue is actively shaking hands with our T cells and paralyzing them.
Exactly. It's an active suppression.
That completely changes how I view age-related immune decline. Does the spatial infiltration explain why immunotherapies, which are designed to wake up exhausted T cells, so often fail in older breast cancer patients?
It provides a very compelling mechanistic explanation, yes. If the fundamental architecture of the aging tissue is already expressing FAM3C and chronically binding to PD-1 before a tumor even initiates,
then they're already exhausted.
Right. By the time a tumor actually forms, those local T cells have been deeply exhausted for months or even years. You are administering an immunotherapy drug into an environment that has already established a rigid, localized immunosuppressive network.
Wow. So summarizing this total systemic breakdown for recap number 3, the immune system's fresh recruits drop. They're replaced by memory T cells that migrate directly to the aging ducts. But once they arrive, the aged duct cells use specific surface proteins to physically dock with the T cell's exhaustion receptors, actively putting the security force to sleep.
Perfectly said. And this leads us to the ultimate reality check of the study. All of this incredibly detailed single-cell choreography was observed in mice.
Right.
The fundamental question is whether this localized active aging process translates to human biology.
Because if it doesn't, it's just a fascinating quirk of a rodent. Did the researchers look at human tissue?
They did. They took the distinct genetic signatures of the aged mouse cells, the specific genes that turned on and off, and cross-referenced them with the Cancer Genome Atlas.
Oh, the TCGA database.
Yes. Specifically, they were looking at human patients with luminal A and luminal B breast tumors. These are the most prevalent subtypes in older human patients.
So they compared completely healthy aged mouse cells directly to cancerous human tumors. What was the overlap?
The overlap was staggering. The specific genetic shifts driving the mouse tissue into an aging state perfectly mirrored the shifts driving human tissue into a cancerous state.
Wait, perfectly mirrored?
Yes.
Oh.
For instance, in the epithelial cells, both the healthy aged mouse and the human tumor upregulated MX3A, which is a gene that drives cells toward an undifferentiated stem-like state.
Losing their identity.
Exactly. Both upregulated CXCL17, a signaling chemokine heavily associated with poor prognosis and tumor progression. And crucially, both the aged mouse cells and the human tumor's epigenetically silenced FNDC4, completely dropping that anti-inflammatory shield we discussed earlier.
Yeah, that is eerily similar. What about the fibroblasts? The stroma?
The human tumors display the exact same structural corruption. The human cancer stroma matched the aged mouse stroma by heavily upregulating FAP, that cancer-associated fibroblast marker.
Unbelievable.
And in the immune compartment, the human tumors relied on the exact same exhaustion checkpoints, PD-1, GZMK, and LAG3, that the aged mouse tissue used to put its immune cells to sleep.
So the mechanism is virtually identical. A cell in a healthy aging mouse looks, behaves, and signals alarmingly like a cell inside a cancerous human breast tumor.
It does.
Which forces a really uncomfortable question. If aging is essentially creating a pre-neoplastic state across the board, rewiring the genetics, mutating the infrastructure, and actively putting the immune system to sleep, does the concept of a healthy aging breast even exist?
Well, from a purely clinical perspective, yes, because there is no malignant mass threatening the patient's life.
Right.
But from a molecular perspective, this study demands that we redefine what healthy means. The tissue is not healthy in the sense of being youthful or stable. It is existing in a state of high wire tension, where the biological soil has been fully terraformed to welcome a malignancy. The risk is woven into the physical architecture of the tissue itself.
So to bring all of these complex mechanisms together into one final picture for you, we set out to understand why getting older is the #1 risk factor for breast cancer. What this Nature Aging Atlas reveals is that aging is an aggressive, coordinated assault on the tissue's identity.
Yes.
The epithelial cells undergo an epigenetic crisis, shutting down their anti-inflammatory shields.
Yeah.
Their distress signals panic the local fibroblasts, causing them to abandon normal construction and build mutant, tumor-friendly scaffolding.
Yeah.
And when the immune system rushes in to help, the aged tissue physically docks with the T cells and exhausts them into submission. The red carpet isn't just rolled out. It is painstakingly woven, thread by thread, year by year.
And by utilizing technologies like single-cell RNA sequencing and spatial mapping, we finally have the literal blueprints of that red carpet. We can see exactly which genes are driving the transformation.
Which leaves us with a truly fascinating idea to consider. If aging creates this localized, contagious environment of dysfunction where stressed cells trick the surrounding structural and immune cells into a cancer-friendly state, does this apply to other interventions we undergo?
That's a great question.
Right. Think about it. If someone gets a cosmetic, skin-deep laser treatment or chemical peel designed to artificially induce acute stress and force dermal fibroblasts to rejuvenate and pump out new collagen, could that artificial signaling accidentally be altering the long-term cancer risk profile of the tissue beneath it?
Oh, wow.
Or expanding outward, is this exact same active suppression happening in the brain with Alzheimer's? Are aged neurons physically putting microglial immune cells to sleep decades before the first amyloid plaque ever forms? The microenvironment is everything, and the older it gets, the more it seems to have a mind of its own.